The first document known to mention descriptions of a disease resembling diabetes is an Egyptian papyrus dating from approximately 1500 BC. Diabetes mellitus is the general term for metabolic diseases characterized by hyperglycemia and is caused by defects in insulin secretion, insulin action, or both. Type 1 diabetes. Type 1 diabetes is an autoimmune disease that commonly appears in children. It is a chronic disorder in which the immune system targets the pancreas and destroys insulin-producing cells, leading to little or no insulin production. About 10% of diabetes cases are type 1 diabetes cases. If severe enough and untreated, type 1 diabetes can lead to amputation of feet, coma, and even death. Type 2 diabetes. Type 2 diabetes is characterized by impaired insulin action and/or abnormal insulin secretion. Several genetic defects have been linked with the disease, and these are inherited in an autosomal dominant pattern. Most are mutations in transcription factors, which are involved in the tissue-specific regulation of gene expression in the liver and in pancreatic beta cells by binding to specific segments in the DNA. One of these mutations involves glucokinase, which is an enzyme which catalyzes the phosphorylation of glucose to glucose 6-phosphate. The main cause for type 2 diabetes is defective post-receptor insulin signaling. Glucose enters the cell through facilitated diffusion with the help of the transport protein GLUT2. The enzyme glucokinase then adds a phosphate group to the glucose molecule in glycolysis. The generation of ATP from cellular respiration leads to the closure of the potassium ion channel. Binding of the sulfonylurea receptors controls this action. High levels of glucose leads to increased production of ATP, which in turn binds to these receptors. This channel leads to depolarization of the plasma membrane and influx of positively charged calcium ions. The difference in charges then triggers and allows insulin to be secreted. GLP-1 on the outside of the cell senses glucose levels and in turn enhances insulin secretion. Without enough insulin, the body is unable to use sugar for fuel, causing it to break down fat instead to use for energy. Waste products called ketones and non-esterified fatty acids are produced when the body breaks down fat. These free-flowing radicals impair beta cell function and lead to insulin resistance. Furthermore, hyperglycemia or increased glucose levels induces apoptosis or rapid cell death in fragile beta cells, which can worsen the person's condition. Thank you for listening to our presentation, and special shout out to Mrs. Capolino and Jason for committing to UPenn, baby. Let's go!